everyone. In this video, we're going to look at what enzymes are, how they work, and some of the factors that can affect them. If you haven't seen it yet, I recommend pausing this and watching the video on metabolism before going further. Enzymes are biological catalysts, usually proteins, which help to speed up chemical reactions within cells. You can usually tell an enzyme by its name. It'll usually have ASE on the end, and the rest of the name will refer to the substrate or function. For example, sucrase is the enzyme that breaks down sucrose, lactase breaks down lactose, and DNA polymerase makes a polymer of DNA. Regardless of the specific reaction they work on, all enzymes speed up reactions by lowering what we call activation energy, the amount of energy needed to get a reaction going. If you think about a car, assuming it has gas in it, it has plenty of energy to power its movement, but it doesn't actually go until you start the ignition. The ignition is the activation energy. Let's consider a reaction where we're going to start with a reactant with a certain amount of free energy. Before we can break that into our products, we have to put in some energy to make the bonds in the reactant less stable. And the difference between this and where we started is the activation energy. Once we've done that, we can release energy by breaking the less stable intermediate into the products. Performing the same reaction with an enzyme, shown by this orange line, doesn't alter the amount of energy that we get out because the reactants and the products are the same. The enzyme is able to speed up the reaction by lowering the amount of activation energy. This could be done by contorting the bonds in the reactant, providing reactive functional groups, or other mechanisms that decrease the amount of energy required to make the reaction happen, such that the temperature within the cell will then often provide sufficient activation energy for the reaction to occur. Let's have a look at how enzymes work. As catalysts, enzymes speed up a specific reaction, but aren't used up by that reaction. So once it's complete, the enzyme will be free to start over again. Our enzyme, shown here in green, has a specific region called an active site, which is where the substrate, the molecules that the enzyme will work on, will bind. The substrate will enter the active site, causing the enzyme to change shape and close around it in a model that we call induced fit, similar to how your hand closes around another person's during a handshake. This brings the required functional groups into the correct positions, leading to catalysis and release of the products, returning the enzyme to its original shape, ready to pick up another substrate molecule. Like everything else in a cell, it's important that enzymes are controlled, and there are different ways that the rate of reaction can be regulated. First, there are molecules called inhibitors, which, as the name suggests, prevent enzymes from catalyzing reactions. These inhibitors can be classified into one of two broad types, competitive and non-competitive. A competitive inhibitor is so called because it competes with the substrate for the active site, physically blocking it from getting to the enzyme. A non-competitive inhibitor, on the other hand, binds away from the active site to what we call an allosteric site. So the active site is still available for the substrate, but the inhibitor typically alters the shape of the enzyme so that the active site no longer functions effectively. This is what happens in negative feedback, or feedback inhibition, where the end product of a metabolic pathway often functions as an allosteric inhibitor of an enzyme at the beginning of the pathway, thereby limiting its own production. Temperature can also affect the rate of enzymatic reactions. At low temperatures, molecules move very little, and so there are limited interactions between enzyme and substrates within the solution. As the temperature increases towards optimum, the molecules move more, increasing the number of enzymes and substrates that interact, and so increasing the rate at which the reaction occurs. In many organisms, like humans for example, this is around 37 degrees Celsius, but the exact optimal temperature will depend on the organism. For example, in thermophilic bacteria that live in hot springs, the optimal temperature is closer to 75 degrees Celsius. If the temperature increases too far beyond the optimum, the enzymes will denature, as the bonds stabilizing the tertiary structure will break, and the rate of reaction will drop very quickly. This is also the case with pH. Deviation too far from optimal will cause denaturation of the enzymes and a subsequent drop in reaction rate. Now, this doesn't mean that all enzymes work best at neutral pH. However, many do. But others function in specific cellular compartments, like lysosomes, or even specific organs, like the stomach, allowing the pH to vary from one place to another. In addition to inhibitors, temperature, and pH, other factors can also affect the rate of reaction. Increasing the substrate concentration, for example, can increase the rate of reaction, but only up until all of the active sites are occupied, which we refer to as saturation. At this point, the only way to increase the rate further is to increase the concentration of enzyme. Many enzymes also require additional components called cofactors, which are inorganic like metal ions, or coenzymes, which are organic. In fact, many of the vitamins that you need in your diet are coenzymes, or precursors that they can be made from. In these cases, the cofactor or coenzyme will bind to the enzyme, along with the substrate, to assist in catalysis. Thanks for watching, and if you haven't checked it out yet, please go and check out the video on metabolism.